Hello and welcome everyone to BioKuria 2021. My name is Sung Park. I'm an Associate Manager of Strategic Planning and Business Development Office of Nexel. On this presentation, I'm going to give a company introduction of Nexel here. Nexel specializes in iPSC or induced pluripotent stem cell technology. We spinned off from Korea University in 2012, and we're the first Korean company to license iPSC technology from iPS Academia Japan. Nexel has a history of rigorous R&D. Ever since we spinned off from Korea University in 2012, we launched 3 cell products named hepatocytes, cardiocytes, and neurocytes, which will be explained in detail on the later slides. And we also launched our cardiotoxicity screening assay called Next Service. Along with the iPSC technology development, we also have been developing new drug pipelines called MP11 and FPT0025. On this company intro, we'll be majorly focusing on 3 major branches of Nexel business. The first part will be iPSC-derived cell business. Second part is the cardiotoxicity screening assay developed by Nexel called Next, and the new drug pipelines that we are currently developing. The first part is Nexel's iPSC-derived cell business. The subsections of this part would be what are induced pluripotent stem cells, why use induced pluripotent stem cells, and how Nexel specializes in differentiation of iPSCs, and the officially marketed product named cardiomyocyte S. So before we jump into how Nexel is specialized in iPSC technology, we need to look at what the induced pluripotent stem cells are. In a brief definition, adult cells are induced to become pluripotent. This idea was awarded a Nobel Prize in 2008 in Physiology of Medicine. And on this diagram, we can see a brief concept of iPSC making process. An adult cell from your body is taken off, such as hair cell, skin cell, any cell from your body, and it is reprogrammed using the iPS reprogramming factors. And the adult cells are reprogrammed into iPS cells, which have a huge ability to potentially divide into any types of somatic cells, such as cardiac muscle cell, red blood cell, liver cell, skin cell, and so on. So with this huge potential to become any types of cells, we can apply them to many areas. These application areas are subdivided into 3 major areas. The first area would be cell therapy using iPSCs. Let's say if there is a damaged organ that cannot be treated with the current therapeutics easily, we can apply an iPSC-derived cell transplantation to turn the damaged organ into a healthy state. Or in a drug screening application, normally to treat a disease, there are multiple types of drugs available on the market, and to choose the most efficient drug or the most effective drug, we can screen them on the iPSC-derived cells and then choose the most effective drug for your disease. The third major part would be the in vitro research, since iPSC research has a large area to be discovered. iPSCs and iPSC-derived cells are used in diverse fields of research, and the examples could be safety study, toxicology study, modeling study, and so on. So in order to perform these large areas applications, there should be a core technology to use the iPSC, and that is the differentiation technology. Nexel is the specialist in differentiation of iPSCs. Our technology is manifested in 3 marketed products named neurocyte, which is iPSC-derived neurons, hepatocytes, iPSC-derived hepatocytes, and cardiocyte, iPSC-derived cardiomyocytes. The major focus of this presentation would be on the cardiocyte S, the cardiomyocytes that is officially marketed in Nexel. Cardiocyte consists of 5 million cells in frozen state, and it is provided with 200 milliliters of cardiocyte S media. The strengths of cardiocyte S product comes from the similarity with the human cardiomyocyte. Cardiocyte S product has high functional similarity and high morphological similarity with the human primary cardiomyocytes. As you can see on this picture, this is the microscopic image of cardiocyte S. You can see the morphological similarity with the human cardiomyocyte. Also, on the video above, you can also see the beating action of our cardiocyte S product which shows the high function similarity with the human cardiomyocyte. Our iPSC drug cell business application is not limited to the cardiomyocyte or cardiocyte S. We also have the market available products called hepatocyte, which is iPSC-derived hepatocytes, and neurocyte, which is the iPS-derived cortical neurons. And we aim to further our research into the insulin patch, spinal injury treatments, and cardio patch, and so on. And what's important to note is that we already developed application of the cardiomyocyte research, and it is called Next Cardiac Safety Screening Assay, which will be explained in detail in the second section of our presentation. The second part of our business area is the Next Cardiac Safety Screening Service. The subfields of this topic would be the importance of cardiac safety, next cardiac safety, the comparison between HERG assay and the next protocol, and the ICH guideline revision that Nexel is currently participating on. Before we jump into how next protocol is developed by Nexel, we need to know the importance of cardiac safety in drug development. As we all know, a tremendous amount of time and money is invested into drug development, and the survival rate is very low. And we need to note that cardiotoxicity is the leading cause for drug attrition, and 45% of drug withdrawals are related to cardiovascular issues. There are real-life examples of this, such as dimethylamine, propoxyphene, and rosiglitazone, and these drugs were withdrawn after the cardiotoxicity-related death. So we need to know that before we jump into the actual drug development and investing huge amount of money and time, we need to screen for the cardiac toxicity of a drug preemptively. So what does it mean by that the drug is cardiotoxic? It generally refers to a pro-arrhythmic property of a drug. There's a regular periodic heartbeat that our heart normally has. And when a pro-arrhythmic drug is treated, a torsade de point occurs, also called a TDP, and it turns a regular heartbeat into irregular patterns of heartbeat. And this irregular patterns of heartbeat leads to a heart attack or sudden death, and this is why we need to look at the cardiotoxicity of drugs. So a TDP, or torsade de points, occurs due to QT prolongation. QT prolongation occurs in the patient level, and when we look it into cell level, it is called the action potential prolongation, and this is caused by the ion channel blockage in the protein level. Next protocol service looks at these potential dangers of the cardiotoxicity of drugs. And our next cardiac safety screening service is performed by using our cardiomyocyte product called Cardiocyte. And we combine it with the available MEA assay machines and then screen for the cardiotoxicity of your drugs. The services could either be the screening service, which is high throughput of library test, and that looks for the single dose hit detection. And it could be a safety pharmacology of a single molecule, and we can screen for the dose response to cardiac safety of your molecule. Next, cardiac safety looks at various parameters. Here you can see the beat rate, contraction amplitude, rising time, falling time, and field potential duration. And the ECG generated by Next service is highly similar to the healthy human ECG, which could lead to more precise and accurate results of the cardiac toxicity screen test. And here you can see the comparison between the HERG assay and the Next protocol. HERG assay is the current method for screening cardiotoxicity of drugs. It has about 70% accuracy, low specificity, which means it can have false positive or false negative. It only looks at the one ion channel of the cardiomyocytes, and it is low throughput. On the Next protocol, we try to enhance the performance of the safety screening assay. It has accuracy higher than 87%. It has high specificity, and it looks at multiple ion channels available on the cardiomyocytes. And it is very efficient by doing high throughput assays. Nexel's efforts to enhance the cardiac safety protocol is also manifested in our effort to revise the ICH guideline. there's a cardiotoxicity guideline available on ICH called S7B guideline. And this guideline is under revision by the governmental entities such as FDA, Korean FDA, PMDA, and EMA, and so on. In association with the research group HESI, Nexel belongs to this research group to research the cardiac safety screening assay. So far, we looked at the iPSC technology of Nexel and its application, Next Protocol. And we're at the third part of Nexel's major business area, which is new drug development. The subfields of this topic would be the history of our drug development, the major pipelines profile, its main target models, and the development status. To start off with the history of our drug development, our drug development actually started with the iPSC research. In 2012, we transplanted iPSC-derived hepatocytes in the liver of the mice and observed the fibril area regeneration. From these findings, we analyzed the iPSC-derived hepatocyte secretome, and they figured out that there's a protein called MFGE8, and that it has an anti-fibrotic function. From these findings, we recombined the domains of MFGE8 and discovered our lead pipeline called MP11. And here's the brief profile of MP11. As described earlier, the origin of MP11 is the secretome of human iPSC hepatocytes and is a recombined version of MFGE8 protein. And since it is a naturally derived protein from human body, it has no toxicity up to 350-fold of working dose. Its size is 22.6 kilodaltons, and the target indications is fibrosis, and specifically in liver fibrosis and myocardial infarction. The GLP toxicity study is near completion, and the clinical phase 1 is expected in second half of 2021. The antifibrotic property of MP11 comes from its unique mechanism of action. As you look at this diagram, fibrous progression starts from liver injury, and it activates the hepatic stellate cell, and it causes the fibrosis of the liver tissues. MP11 inhibits this progression by downregulating TGF-beta expression and signaling, and inactivates the hepatic stellate cells. MP11 also interacts with macrophage and enhances collagen uptake and provides optimal environment for proper liver regeneration after liver fibrosis. The MP11's main target indication is fibrosis, and specifically liver fibrosis and myocardial infarction. Liver fibrosis results from chronic liver damages. It produces a scar-like and fiber-like state, and if left untreated, it develops the cirrhosis or cancer. MP11 is treated in the fibrotic stage of this disease progression, stops the progression of the disease, and then provides an optimal environment for regenerating the liver tissues. In myocardial infarction, it results from epicardial vessel blockage or physical shock, and it is also known as the general term heart attack. When the heart attack or MI occurs, the cell dies in this infarcted area, and the myocardial activity is left weakened even after surgical treatment of this heart. And MP11 is treated in this infarcted area to treat the fibrotic state, become healthy again. Here's the preclinical data for the MP11's efficacy in liver fibrosis. As you can see in this photo, this is a normal state of mice liver. We induced the liver fibrosis with the TAA molecule, and here you can see the red stained area, which is fibrotic. When we treated MP11 in the TAA-induced model, we can see that the red stained fibrotic area has been decreased significantly. When you quantify this result in pixels, you can also see on this graph, you can see a significant difference of the fibrotic area pixels between the TA-induced model and the MP11 treated model. Summary, MP11 treatment reduced fibrotic and fatty area in liver fibrosis model. And here's the preclinical data for MP11's efficacy in myocardial infarction. This is the red heart, and we induced the myocardial infarction. And you can see the blue stained area of the infarcted tissue. When we treated MP11, you can see a decreased area of infarcted tissue, as shown here. And we also noticed a enhanced cardiac activity when we treated MP11. In the summary, MP11 reduced infarcted area and enhanced the cardiac safety in a rat myocardial infarction model. So here's the development status of various indications of MP11. Liver fibrosis and myocardial infarction is near completion of the GLP package of preclinical development, and the myocardial infarction is expected to enter the clinical trial phase 1 in second half of this year. We have other indications such as non-alcoholic steatohepatitis and severe alcoholic hepatitis, and we are also developing a prognostic marker for SAH, and it is under clinical trial phase 1. So we are nearly at the end of our presentation. So here's a summary of the presentation, what to expect now and next from Nexel. First, Nexel is actively growing iPSC-derived cell business, both locally in Korea and globally. Also, we have started to operate as a service provider for testing cardiac safety. And for MP11, our major pipeline, we're looking for open innovation or license out opportunities. Thank you for listening. And if you have further questions or comments, please feel free to contact us. Thank you.